quantities of DNA. We're using all of the same techniques the medical examiner's office uses. In fact, my former postdoctoral assistant and 3 of my former grad students work just down the street at the medical examiner's office, applying these genetic techniques to identifying criminals, from things as small as the fingerprint left behind on the trigger of a gun, actually placing a person not only with the weapon but with his hand on the trigger, because a cell or two is sloughed off and that will leave a DNA signature behind. We are exploring different aspects of the genome. Our genome is this incredibly interesting and intricately complex amalgam of all sorts of things. We have ancient dead viruses in our genome that have actually been, we were once infected with them tens of millions of years ago. They are now dead, but their DNA actually has crept into our genome. There are jumping genes and repetitive DNA. The chromosomes themselves can fission and fuse. We can look all the way down to the DNA base level, the A, C, Gs, and Ts. In our lab, we're taking advantage of all of these different aspects of the genome to try to figure again out evolutionary relationships. So we can come up with basically an entire tree of the primates, and from that we can get at more and more interesting questions. So almost all of my work, we occasionally work on non-primates, but most of the work in our laboratory has focused on monkeys, apes, lemurs, and lorises. As I said, this work has now become much easier. Way back in 1989, we had to manually sequence DNA. We had to carry out 4 separate test tubes worth of reactions for each little bit of sequence that we gathered. We had to use radioactive substances and X-ray films to visualize the results. We had to count up these results just by following one's finger. This is an ancient, i.e. 1990 DNA autorad, in which you have your A's, C's, G's, and T's, and we used to have to go say G, C, C, A, G, G, T, A, and write that down to work this out. I managed for my PhD thesis, which I received in 1992, to collect 684 bases of DNA from 13 different species of animals. That's today a tiny amount. Now my students can collect between 500,000 and 1 million bases of DNA in our laboratory alone. And if you go to one of those large genome centers, even more. These technologies have moved as fast and as far as computers have went. So we went from having to look at the individual A's, C's, G's, and T's to being able to mark them with different colored dyes so we could look at them all at once with a different color representing them, to the point now we actually run these things through microscopic capillaries, shoot a laser through it, and can actually detect the individual DNA bases as they migrate by, what color they are, and we can do this 48 or 96 or even 384 samples at a time, and in a matter of an hour or two, generate thousands of bases of DNA from each of those single capillaries. So again, we can now do 1 million bases of DNA a day in our laboratory, and that's without breaking a sweat. I mean, if we really, really tried, we could even up that. And that's just from this single machine here. Imagine if you had a whole factory full of those machines. We can now sequence a complete genome, all 3 billion bases, in less than a week's effort in one of these major genome centers. And that's why we're getting so many of them. One of the genomes, though, that we focus on in our lab is actually a special part of our genome called the mitochondrial genome. In almost all of your cells, you have hundreds of these little organelles, the mitochondria, that contain their own genome, That's about 16,500 bases long. And because there are hundreds of these, and in fact thousands of copies of this genome in each and every one of your cells, it's very easy to work on in the lab. It also has the nice feature that it is purely maternally inherited. And so you can get a very clean evolutionary history of your mother's mother, and so on, all the way back. And so a lot of our effort has focused on the mitochondrial genome. And in fact, one of the projects we're doing this summer with your fellow students in my lab is we're developing computer programs and algorithms to analyze these mitochondrial genomes that we're generating almost weekly in our lab, so that we can carry out additional evolutionary analyses. So I even had one student, an undergraduate, actually sequence 16 complete mitochondrial genomes from baboons to try to see if fetal stress and distress actually affected the mitochondrial genome and then led to problems later in life. But all of these efforts would be wasted without incredibly powerful computer algorithms and approaches. So again, some people, it turns out, just don't have what we call lab hands. You just are klutzy or something. You just don't do lab work very well. There's still so much research you can do, though, on the computer. And we are developing new algorithms, and teams all over the world are developing very sophisticated techniques for analyzing all of this evolutionary and medical data in, again, the field we call bioinformatics. And so it has become the growth field. The last several people that I have sent out of my lab with PhDs all went on to jobs in new fields of bioinformatics. So this is something I want to really sort of preach and strongly encourage you to consider, is learning more about how to analyze the data. Not just setting up an Excel spreadsheet to calculate your means and do some tests, but to really learn how to utilize the vast amounts of data that are increasingly out there. There are public databases now. Every piece of DNA being sequenced and published is available freely in public databases. Just finding the information, it's worse than Google. Finding the information you need takes some very powerful and creative approaches. And so I would encourage you to do that. In my lab, we have to use computer clusters, parallel processing, and all of that to do this. But it turns out you can do many of these things very inexpensively and even on your laptop. On this laptop I have here, I have the complete orangutan, human, chimp, macaque, and gorilla, and 2/3 of the gorilla genome on my hard drive, and I can sit at home at night analyzing those data. So you can do this on that. And in fact, on one of my little jump drives here, I have 4 genomes stored that I was going to bring into work today. Here's an example of a piece of the orangutan genome, for instance, called or pulled off of one of these public databases. There are many, many different ways to search these, to represent these. It's very important as you